Okay, let's go ahead and get started. Okay, are there any questions regarding the last lecture? I'm sure I'll get plenty of emails, which is perfectly fine. It's easier for me to answer that way. I have time to actually look it up in a book and see if I get the right answer. Okay, the last lecture here, we're going to talk a little bit about some cell-to-cell interactions. And the first part, we're going to talk about what is required for the B cell response. I know Dr. Mayer discussed a lot of these different aspects, but what I'm going to try and concentrate on here is a little bit about what are the cell-to-cell interactions that are required. And what we'll notice is that we'll discuss are the T-dependent as well as the T-independent antigens. And what we'll see with the T-independent antigens is that there's a particular cell population known as the CD5 or B1 cells that play a big role in the response to the T-independent antigens. And the last part will kind of put you to sleep by discussing many cytokines and what their role is in the immune response. Okay? Start by talking about the T-dependent antigens. What's meant by T-dependent antigens, it means that in order for the B cell to respond or to produce antibodies, they need T cell help. They can't do it by themselves. And typically, there is what is known as the hapten carrier effect or linked recognition. So typically, the T helper cells will recognize the carrier, where the B cell will recognize the hapten. The carrier typically is a protein. As we discussed before, T cells have to recognize proteins. T helper cell and the B cells cooperate by interacting with one another. And again, interactions are class 2 self-MHC restricted. So the peptide presented by the B cell to the T cell is an association with a class 2 MHC molecule. You probably already went over this, but here's a brief definition of what a hapten is and what a carrier is. A hapten can be any small chemical group that cannot elicit an antibody response on its own. Typically, these are too small to cross-link the B cell receptor, and by themselves, they cannot recruit T cell help. One example is an immune response to penicillin. Typically, most individuals don't form an immune response to penicillin. However, the ones that do, what happens typically is penicillin is broken down, and the metabolic products act as a hapten, will bind to some protein within the individual. This interaction will then lead to the immune response. A carrier is a protein that can be physically linked to the hapten. You have to have the protein and the hapten with each other. Now, the carrier, or the one protein itself, can have both a hapten and a carrier on it. The carrier can carry multiple copies of the hapten. This could allow for cross-linking of the B cell receptor. That's one of the things that's missing in the hapten. They're too small. They can't cross-link the B cell receptor. And also, the carriers can elicit T cell help by inducing the response of that T cell to that protein, to the carrier protein. The B cells are very unique, a very unique cell, in that they express both immunoglobulin receptors and class 2 MHC molecules. They're capable of producing antibodies with the same specificity as what's found on its surface IG. So the immunoglobulins can act not only they can not only be secreted, but they can actually act to bind to and bind to different peptides, leading to presentation of the class 2 molecule. That's shown here again. They can produce immunoglobulins that produce a particular part of the immune response. They can also act as antigen-presenting cells. Typically, the haptens are recognized by the immunoglobulin. So the immunoglobulin is able to recognize that small chemical part of the carrier hapten complex. Both the hapten and the carrier will then become endocytosed. The whole complex will be engulfed by the B cell. The carrier part will be processed and presented to the T cell. So the carrier part is the part of the peptide or part of the antigen that will be chewed up and presented with the MHC class II molecule and then recognized by the T cell. Once activated, the T helper cell will then produce the cytokines. The cytokines will then help to stimulate the B cell to produce the immunoglobulins. There's two parts here. You have to have the ability of the B cell to somehow recognize the hapten. That leads to the engulfment of that hapten carrier complex. That becomes basically chopped up and presented with the MHC class II molecule. And the T helper cell will recognize the part of the carrier molecule, and that will lead to activation of the T cell, production of cytokines, and then the subsequent activation of the B cell. And that's shown here in the cartoon. So we can go through the steps here. You have your antigen. This can be your hapten carrier complex. You have a B cell that expresses the immunoglobulin specific for the hapten part of this complex. This becomes processed and presented. So the hapten part is what becomes recognized. Your processes and presentation of this whole complex. Typically, a different part of that complex will be presented with MHC class II. The carrier part will be presented with MHC class II. This will lead to the expression of the B7 by the B cell. Once that's expressed, CD28 on the T helper cell will lead, this interaction here will lead to the second signal. Again, as we mentioned before, that in addition to this interaction between the APC and the T helper cell, you have to have co-stimulation. So that happens with the B7 and CD28 interaction. The T cell is then activated. Upon activation, it can produce cytokines. In addition, it upregulates a ligand, a CD40 ligand. That binds to the B cell. This acts kind of as a second signal to the B cell as well. The combination of the cytokines and the interaction with the CD40 ligand on the B cell then leads to the complete activation of the B cell and production of the immunoglobulins. Just to show it to you one more time, again, antigen of the carrier, the hapten, interacts with immunoglobulin. It becomes processed and presented with the MHC class II molecule. That is then presented to a T helper cell, leads to upregulation of B7 on the B cell, which these two signals here, first signal, second signal, lead to activation of the T cell. This activated T cell then produces cytokines as well as CD40 ligand. CD40 ligand binds to CD40 in the B cell. The combination of CD40 ligand with the cytokines then leads to full activation of the B cells, and they can then produce the antibodies. And the question that came up during the break was, what is the difference between, when you talk about thymic education, the T cell is taught to recognize self from non-self. So they don't recognize self. The B cell, I guess in a sense, can recognize pretty much anything. It doesn't have the same restriction. So what's to keep the B cell from producing antibodies against self? And typically, these B cells require help from a T helper cell. So unless there is foreign antigen that is recognized by the T helper cell, you will not get antibody production by these types of B cells. So that's a way in which the T helper cells can regulate what antibodies are being produced by the B cells. That make sense? You have the question. You can't ask it again. No, go ahead. Initial step here? Here. What happens here is that this B cell is basically unactivated, circulating throughout your blood. It will come into contact with the hapten carrier. The hapten part of this will bind to the immunoglobulin, and this will then be brought into the B cell. This complex will be processed and presented. So up to this step here, the B cell is simply acting as an antigen-presenting cell. Further down the line, it also becomes activated and produces immunoglobulins. Now, in addition to that, there are other ways that the B cell can be activated. And again, this still requires T cell help. But in this case, as we mentioned before, there's more than one type of antigen-presenting cells. B cells aren't the only cells that can act as antigen-presenting cells. We also have dendritic cells and macrophage. So in this case, you have a T helper cell that recognizes a MHC class, recognizes an antigen in association with MHC class 2. This cell becomes primed, so it becomes semi-activated. It's able to, some of the co-stimulatory molecules such as CD40 ligand may be upregulated. This cell then can go out into the periphery and interact with a B cell that has processes and presented the carrier part of this. So once this primed T cell interacts with this B cell expressing the same antigen that it was presented by the other APC, this can deliver the appropriate signals to this B cell to then produce the antibodies. So instead of the B cell presenting the initial class 2 molecule association with the antigen, a separate one does, produces a primed T cell. This primed T cell then is able to help the B cell by producing cytokines and delivering the signals. This leads to can lead to antibody production. Initially, it leads to division of the B cells, then to production of antibody by the antibody-forming cells as well as memory B cells. So is that sure? Back. This one? Once this signal happens, typically, once this interaction happens here, this will send a signal to the B cell to upregulate the B cell. Okay. It turns out that B cells can play a very important role in the secondary immune response. We'll discuss that a little bit here. As I mentioned in the previous slide, in addition to producing antibody-forming cells, memory cells are formed. Okay. These memory cells have very high affinity receptors, so they're able to bind antigen to very high affinity. Therefore, it takes very small quantities of the antigen to activate these cells. So it takes much lower concentrations of the antigen for the B cell to be able to pick it up and then present it to the T cell. Now, this is very important in a secondary immune response because typically what you see, once you've been exposed to an antigen the second time, the response is much quicker. And the reason it's much quicker is because it takes less of that particular pathogen to start the immune response. So instead of 6 days into an infection, your immune cells are seeing enough antigen. As soon as you're infected with that particular pathogen, the B cells are able to pick up those antigens and then present them to the T cells and start the whole immune response. That makes sense? Okay. Now, in addition to the T-independent or T-dependent antigens, there are also T-independent antigens or thymus-independent antigens, and these antigens do not require the help of the T cells. These are very unique. Again, they can activate B cells without class 2 MHC-restricted T cell help. Typically, it takes a lot of these different antigens, very high concentration of these antigens to elicit the B cell response. And typically, these T-independent antigens are large polymorphic molecules with repeating determinants. So one of the things about the T-dependent antigens was one of the reasons they couldn't activate the B cells because they couldn't cross-link the B cell receptor. By having much larger molecule or antigen with repeating determinants, the chances are that you can actually cross-link the immunoglobulins on the B cell. By cross-linking, you can lead to the stimulation of the B cell. Again, they're poorly degraded. That basically feeds back into this. Of not being degraded, they remain large, and they remain capable of cross-linking the B-cell receptor. Just say another point here. Some can activate both immature and immature B cells, and some can activate just mature cells. And typically, what they found is that the main type of B cell that can respond to T-independent antigens is known as a CD5 B cell or B1 cell. And we'll discuss a little bit about these cells in the next couple slides. And when you look at T-independent antigens, they can be divided into two groups, the TI1 and the TI2. TI1 are predominantly bacterial cell wall components. So if you have a large infection with a bacteria, these B cells may be quickly activated. The T12 antigens are predominantly large polysaccharide molecules with repeating antigenic determinants. Examples are flagellin, that's a part of a bacteria, and maybe different parts of different viruses. Again, the point here is that it takes large concentrations of these antigens to produce this response. And typically, this response is elicited by a CD5 B cell or B1 cell. And if you look at the CD5 cells versus the typical B cell, these B cells are the first ones that appear during development, more immature B cells. They're different in that they express IgM with little or no IgD on the surface. They produce mainly IgM from unmutated or a minimally mutated germline. So the diversity of the types of antibodies they produce is less. These antibodies have low avidity, and they can bind multiple different antigens, so they're polyreactive. So they're very different from the conventional B cells, and the antibodies they produce are also very different. And these contribute most of the IgM in the adult serum. Important part here is that they do not develop into memory cells. That's important because, again, the secondary response typically, or the memory response, is much stronger than the primary. And one control over that is the help of the T helper cells, which really makes sure that you're not recognizing self-determinants. There's no memory cell response here. Again, that's important because there's no T help here either. This is just basically restating this first point here. The secondary response typically resembles a primary response, again, because there's no memory. Interestingly here is that these cells are self-renewing. That means they can proliferate in the periphery and form new CD5 cells, whereas the regular ones are, as we discussed, come from the bone. They're basically generated and mature in the bone marrow, or generated from the bone marrow. You find a lot of these residing in the peripheral tissues, whereas B cells typically you can find in the secondary lymphoid organs. Again, this is kind of reemphasizing this. These types of B cells are the predominant lymphocyte in the peritoneal cavity, which is a peripheral tissue. Now, if you want to compare if you want to call these conventional B cells with the CD5 B cells, if you look at development, you see that the CD5 cells occur early in development, whereas the other B cells occur later. If you look at renewal, these are able to self-renew, where these are replaced by the bone marrow. If you look at the production of immunoglobulin, very high for the CD5 cells, lower for the B cells. Typically, you get a lot of IgM produced by these CD5 cells, where the immunoglobulin production is much less, and typically it's IgG for the conventional cells. Important point here is that as Dr. Mayer discussed, most of the immunoglobulins can only bind one type of ligand, recognize one type of antigen, whereas these are polymorphic. They can recognize many different ligands. Another important point when you look at the different types of T cells is that when you look at the development of tumors in B cells, there are a number of different tumors that can develop with B cells. And a lot of times you can actually classify them by their stage in development. CD5 B cells here, both CD5 and the conventional B cells are generated from lymphoid progenitors. Now, if you look at the tumors that can be generated from lymphoid progenitors, it's typically acute lymphoblastic leukemia, and that's found in the bone marrow. Again, that's where the lymphoid progenitors are found. If you look at the CD5 B1 cells, again, they're found in the periphery in the blood, and typically the type of tumor that develops in this is chronic lymphocytic leukemia, more of an immature type. Pre-B cells can form pre-B cell leukemia. Still, pre-B cells are typically found in the bone marrow, and that's where these tumors are found. Once you get into the mature B cells, there are a number of different tumors. One is follicular center cell lymphoma, or Burkitt's lymphoma. Again, these are found in the periphery. B cell IgM secreting cells, a bit further along in the maturation process. These can produce what is known as Waldenstrom's macroglobulinemia. It's a whole mouthful. And once the cells become plasma secreting cells, so the very mature type that are activated, they form multiple myelomas. The point here is that during the different stages of B cell development, you have different tumors that can develop, and they're typically located in different parts of the body. Okay. Let's switch gears a little bit. Let's talk a little bit about cytokines. Now, there are so many different cytokines that it's beyond the scope of this particular course, or practically any course, to try to discuss all the different cytokines and all the different functions. So what I'd like to do here is just kind of concentrate on some of the important cytokines and discuss with you what role they play in the immune response. In general, cytokines, it's a very broad class, but typically they're non-antibody proteins, and they act as mediators between cells. So they basically, cytokines produce, and they could then lead to activation of another cell. Monokines, so if you divide cytokines into broad groups, monokines are these types of substances produced by mononuclear phagocytes. Lymphokines are produced by activated T cells, and typically most of the lymphokines are produced by helper T cells. And within these lymphokines and even the monokines, there are interleukins, and a lot of times you'll see the cytokines termed IL-1, IL-2, IL-3, and that's typically the IL stands for interleukin. These interleukins the term came along a while back when typically thought that most of these cytokines played a role in the communication or the activation between leukocytes, so interleukins. Some of the properties of cytokines. Cytokines can be produced by cells involved in both the innate as well as the acquired or specific immune response. Again, these cytokines, the major role they play is they help to mediate the immune response. We discussed this a little bit about with the B cells. The way the B cells become activated is the T helper cells will produce cytokines. For the most part, the secretion of these is very brief and limited. It's thought that they're not stored or preformed. This is not entirely true. There are some cells, typically NKT cells, it's thought that they may have some preformed cytokines, but we won't talk too much about those cells. Synthesis is initiated by gene transcription, mRNA. Again, the mRNA is short-lived. That goes back to the secretion is brief and limited. And again, cytokines are produced as needed. You don't have a lot of cytokines always circulating throughout your body, because if you did, you'd have a lot of activated cells. And if you have activated cells, that can lead to a lot of immune disorders or a lot of different inflammatory responses. Again, cytokines, the production of cytokines is not limited to one cell type. It can be produced by a number of different cells. The macrophage and the T helper cells tend to be two cells that do most of the production, but a number of different cells can produce a number of different cytokines. And this means they're pleiotropic. Another important point is they can have similar action. They're very redundant. So one cytokine, for example, IL-1 or IL-2, may be able to have the same type of interaction or same type of response as another cytokine. This is important because if you have some type of disorder where one of the cytokines is knocked out, you won't lose that particular immune response. And that's shown here. So if you look at the receptors for IL-2, 15, 7, 9, 4. Now, typically, the receptors for these cytokines can either be trimers or dimers. And you'll notice that a lot of these receptors have common subunits. So, for example, if you knock out the IL-2 receptor alpha, you may lose the ability of IL-2 to produce this response, but IL-15 will still be capable of producing an immune response. And that happens a lot. So you have a lot of these particular receptors have very similar subunits. Now, if you look up here, there is one particular subunit that seems to play a very important role. And if you knock that out, you may have big problems with your immune response. Any guesses on which one that is? One? Yes, the gamma receptor for IL-2 receptor. If you knock that out, you basically lose the ability of IL-2, IL-15, IL-7, IL-9, IL-4. And there actually are disorders in which this happens, and this happens very similar to what you see with the knockout of the RAG gene, where you don't have T cell receptors or B cell receptors. If you knock out this receptor, these people become severe combined immunodeficient. So they become very susceptible to a number of different common pathogens, and they're unable to form an immune response. Another important point here is that the production of one cytokine can influence the production of another cytokine. So if you have, for example, production of IL-2, that particular cytokine may then bind to a particular cell, activate that cell, and that cell will then produce another cytokine. And this leads to a production of a cytokine cascade. And exactly which cytokines are produced, which cytokines in this cascade are produced, then can dictate what type of immune response is developed. So you can have either Th1 or Th2. So the development of a Th1 versus Th2 typically is dictated by the types of cytokines that are produced in a cytokine cascade. In addition, for the most part, what we're talking about is that cytokines produced can lead to the stimulation or the upregulation of immune response. In fact, though, some cytokines that are produced actually can suppress the immune response. One example is IL-10. So IL-10 can be produced. IL-10 interacting with the receptor will actually downregulate the immune response. Again, as shown here. So again, they can exert positive or negative regulatory effects. So just by producing a cytokine doesn't mean you're going to have upregulation. They also play an important role in downregulation. And both upregulation and downregulation play an important role in the proper functioning of the immune response. If you simply have upregulation of the immune response, and it goes on and on and on, you'll have a lot of problems with autoimmune disorders. So you also have to have the capability of downregulating the immune response. And this is, again, saying the same thing. The influences of the cytokines upon one another can be antagonistic. So one cytokine can be produced to stimulate the cell, and later on another one will come along to dampen the effects of the first cytokine. They can be additive, so you can have two kind of acting in the same way. So double the cytokine, double the response, or they can become synergistic. So production of two really can ramp up the immune response. The point we're trying to get to here, there's no really hard, fast rules for these cytokine productions and how they act. It really depends on the combinations of the cytokines and in the situation in which they're produced. Important point here is that they bind to receptors with high affinity. They actually bind with a higher affinity than immunoglobulins. So once they interact with the receptor, they stick there pretty firmly. Okay, so once a cell produces the cytokine, the cell responding to that cytokine can be the same cell, which would mean an autocrine effect. So you can have a cell that becomes activated, produces the cytokine. That cytokine can then activate the same cell that produced the cytokine. It can be paracrine, which means it's affecting a nearby cell. And what you see there is typically with the B cell T cell interaction. T cell can produce a cytokine, B cell becomes activated. Or it can be affecting distant cells. So once it's released into the microenvironment, these cytokines can then travel to other areas and affect the response of other cells. And you see this a lot with, for example, chemokines or particular cytokines that lead to the attraction of other cells. So if you have a cell in a site of inflammation producing these particular cytokines that are important for the migration of the cell, they may be secreted out into the system, find a cell in the periphery, interact with that cell, and basically tell it to come migrate to the site of infection. Here, again, the cellular responses to cytokines are typically slow and require new RNA protein synthesis. That's pretty much the case. There are some exceptions to this rule. Now, again, as we first mentioned, the cytokines can have an effect on both the natural or the innate immune response as well as the acquired immune response. And we'll talk a little bit about the cytokines that influence the innate immune response. And what you also see here is that some of the same cytokines keep coming up in both the innate as well as the acquired. So an important point is that some of these cytokines can act to activate or to regulate both types of immune responses. Their tumor necrosis factor. I'm just going to list here basically the ones we're going to talk about, and then in subsequent slides we'll actually talk a little bit about the details. So again, when you look at the natural immune response, there are a number of very important cytokines. You have tumor necrosis factor, interleukin-1, chemokines. This is kind of a broad category of cytokines. These are molecules that act to influence the migration of cells. Type 1 interferon, interleukin-12, and interleukin-10. So when we look at tumor necrosis factor, this is primarily produced by macrophage. The macrophage can play an important role in the innate immune response. Turns out that tumor necrosis factor plays a very important role in mediating the response to microbes, especially gram-negative ones. It also acts by mediating the recruitment of neutrophils to the site, as well as macrophage. So by being produced, it can have a paracrine effect. In addition, anything that's in innate immune response is basically anything that's not included in the acquired. So, interesting, TNF-alpha can also act on the hypothalamus to produce fever, and fever plays an important role in ridding the body of different bacteria. And also can promote the production of acute phase proteins by the liver. These also play an important role in eliminating microbes. There's also interleukin-1. Again, you see a similar trend here. Macrophage, again, produce interleukin-1. The effects are very similar to TNF-alpha. Now, chemokines, as I discussed before, these play an important role in the migration of cells to the site of infection or inflammation. So these basically, the chemokines will tell cells to travel to the site of infection. Chemokines can be produced by many different leukocytes, as well as different tissue cells. Again, chemokines, there are more than chemokines is not just one particular type of substance, but there are actually more than 50 different chemokines so far that have been isolated. We can't talk about all 50, but what we're going to talk about is just the general properties of chemokines. Again, chemokines will recruit leukocytes to the site of infection. So the primary role of the chemokines is to tell cells to come to the site of infection. And that's, again, said here. It plays a role in the trafficking of lymphocytes. Another class is the type 1 interferon, so we can have interferon alpha or beta. Again, when you're looking at the innate immune response, interferon alpha and beta, especially alpha, is produced by macrophage. Beta can be produced by other cells, including macrophage. Importantly here, one of the main functions of the interferon alpha and beta is they inhibit viral infection or viral replication. I think they'll talk a little bit more about that in your virology section of this course. Also, we discussed before, we mentioned that some of the cytokines can lead to the enhanced production of MHC class 1 molecule. Type 1 interferon is one of those such cytokines. So again, production of interferon can lead to upregulation of class 1 MHC, as well as some of the other molecules involved in the processing and presentation with MHC class 1, which then will help to enhance the acquired immune response. Importantly, both of these activate NK cells. And NK cells, again, are the cells that play a very critical role in recognizing barrier cells, including those that have lost expression of MHC class 1. So the NK cells play a very important role in the innate immune response. I told you I'd bore you. We're going to go through about 20 different cytokines and what they all do. By the time we're all done, I'll see a few heads down, but we'll get through it. Interleukin 12, it's like a broken record. Again, what are they produced by? Macrophage, right? And dendritic cells. So again, dendritic, you'll see interleukin 12 more likely will come up again when we talk about the acquired immune response. Stimulation of production of interferon gamma. And it induces the differentiation of Th cells to Th1. So it's very important in the cell-mediated immune response. And the reason I put it here in the innate portion of this is because it's primarily produced by macrophage, and the macrophage can be sometimes stimulated nonspecifically. Again, that's a hallmark of the innate immune response, lack of specificity. Interleukin 12 can enhance the cytolytic functions of CTLs as well as NK cells. And the final one in this category is interleukin 10. The reason this is put in last is because most of the other cytokines typically lead to an upregulation of the immune response. Interleukin 10 is one of the examples of a cytokine that can actually lead to the downregulation of the immune response. Again, produced by macrophage and Th2 cells. So primarily when it inactivates a cell, it inactivates a Th1 cell. And typically the Th2 cell and the Th1 cells have opposite effects. So if a Th2 cell produces interleukin 10 can lead to downregulation of the Th1 response, which in effect leads to an upregulation of the Th2. Again, this is one example of an inhibitory cytokine. It can inhibit cytokine production by activating macrophage. It can inhibit expression of class 2 MHC and various co-stimulatory molecules. So one of the main functions of interleukin 10 is to downregulate the immune response. Okay, switch gears a little bit, the last few minutes. We'll talk about some of the mediators of the specific immune response. Some of these are the same as the ones we just discussed for the innate. We'll discuss their roles and how they affect the acquired immune response. The big one here is interleukin 2. This is one of the first cytokines that was recognized, and it was believed to be one of the main activators of T cells. So you'll hear a lot about interleukin 2. Interleukin-4, it plays a big role in typically stimulating a Th2 response. It can have some effect on the Th1. Interleukin-5 can interact with interleukin-4 to produce an immune response. And again, interleukin-10 here plays a big role. Interferon gamma, again, this was seen in the innate immune response. It's also seen in the acquired immune response. So let's discuss a little about interleukin-2. This is produced primarily by the T helper cells, a little bit by cytotoxic T cells, but primarily by T helper cells. Again, this was thought to be the main growth factor for T cells, but it turns out that interleukin-2 can also affect a number of different other cells. It's not just specific for T cells. Shown here is an example of that. Again, the T cell is the main producer of interleukin-2, becomes secreted, and then it can actually affect a number of different cells. It can lead to stimulation of the B cells, leads to T cell activation, both the proliferation as well as release of interferon gamma. It increases NK cell activity. It also increases the activation of monocytes. Now, this is a particular interest to me because this is also used for the therapy of particular cancers such as malignant melanoma and renal cell carcinoma. And this is actually used in patients who will inject high doses of IL-2. And by injecting high doses of IL-2, you can boost the response of the T cells and the NK cells. This boost in the response allows these cells then to recognize and kill these particular tumor cells. So it plays a very important role in the regulation of the immune response. So again, it can act on a number of different cells, but it can also act in an autocrine fashion. Autocrine basically means the cell that produces the cytokine is the same cell that becomes activated. Shown here is the very simplified interaction between the activated T cell and the MHC class II presenting the peptide. Leads to activation of the T cell, production of the various cytokines, IL-2, 4, 7, all of which combine to similar receptors. Leads to the division of the cell and activation. And eventually, you get downregulation. In this case, you can see downregulation of this immune response by receptor decay. So these cells, as long as they're activated, will continue to produce cytokines, IL-2. You'll continue to see more proliferation of these cells, a rapid expansion of this particular cell. And eventually, this immune response has to be degraded, has to come down. The way it was put to me is if you didn't have regulation or downregulation of the immune response, everybody in this room would be one huge lymph node, because all you have is a bunch of T cells. So what happens is, one way in which it's downregulated is you have the receptors degraded. The cytokine's produced, but it has no receptors. So without the receptors, no response. Last few minutes, we'll talk about the remaining cytokines. Interleukin-4 is another very important one. This is produced primarily by Th2 cells. So these are cells involved in the humoral immune response, or the production of antibodies. This is one that can produce the class switching to IgE isotype. We talked a little about that. Yes? Yeah, Th2 cells are in the acquired, or they're not in the innate immune response. Now, they can actually produce cytokines that can stimulate the innate, but typically they're associated with the acquired immune response. So again, interleukin-4 is produced by Th2 cells, and it leads to the stimulation or the development of Th2 cells. So IL-4 is very important in the development of the Th2 response. And this is basically restated here, promotes growth and differentiation of Th2 cells. Now we'll see interleukin-5 is one of the last ones. Again, this is produced by Th2 cells. In addition to being produced by Th2 cells, one of the main functions of this is to promote the growth and differentiation of eosinophils. So this can activate mature eosinophils. What happens is, one example of how this works in concert with another cytokine is if you have IL-4 produced by a Th2 cell and IL-5 produced by a Th2 cell, IL-4 can activate the class switching to IgE. IL-5 can activate the eosinophils. So if you have high production of IgE and activation of the eosinophils, this can effectively combat this particular infection, helminth infection, particular worm parasite. So simply, this is just an example of how some of these cytokines can act together to produce the optimal immune response. And interleukin-10, again, not only is it important for the innate, it is also very important for the acquired. This is produced by activated macrophage and Th2 cells. This inhibits the production of interferon gamma by Th1 cells. And this interferon gamma is needed to activate the macrophage. So it's kind of a way to downregulate the Th1 response by downregulating the macrophage. And finally, we'll talk a little bit about interferon gamma. This is produced by T helper cells, can be produced by cytotoxic T cells as well as NK cells. This particular cytokine plays a number of roles in the antibody in their immune response. It's typically thought to be a Th1 cytokine. But if you look here, again, this big soup here kind of gives you an example of what this cytokine can do. So it can be produced by NK cells, Th1 cells, as well as cytotoxic T cells, and then activates a number of different cells. Primarily, these responses are Th1 responses. So that's the main. I really want you to know about the interferon gamma. You don't need to know exactly everything that's being activated by the interferon gamma. Just know it's primarily produced by Th1 cells, and it helps to activate the Th1 response. Okay, in addition to activating the innate as well as the acquired immune response, the last couple minutes here, we'll discuss this last slide. Some of the cytokines can actually play a very important role in hematopoiesis, which is the development of the different blood cells. One is known as colony stimulating factor. Again, this plays a very important role in the stimulation of the progenitor cells. So it can lead to the development of a number of the different types of cells. There's granulite macrophage stimulating factor, colony stimulating factor. This promotes, as you can see, the production and maturation of granulocytes and macrophage. There's macrophage colony stimulating factor. As the name says, it's involved in the maturation of monocytes and macrophage. Again, granulocyte colony stimulating factor stimulates production of polymorphonuclear leukocytes, or PMNs. So all these cytokines are very important in the process of hematopoiesis. So in addition to having the cells circulating, a very important point of having a functional immune system is having new progenitors. So these help to generate new progenitor cells. I think we'll end there and pick up tomorrow, the last couple slides. Are there any questions? Yeah.
In the first bullet, what do you mean by bone marrow progenitors?
So a while back we mentioned that all cells of the immune system are developed from the bone marrow, and that comes from a very common bone marrow progenitor cell. This helps to stimulate those cells. So the very, very first step. Any other questions?